We discussed that gray matter was unmyelinated bits, either cell bodies or unmyelinated axons and dendrites. White matter in the spinal cord is going to surround the gray matter. So if you envision the gray matter as that H or that bikini top, the white matter is all the material that's around that. It's going to be made up of our myelinated axons, right? Because again, myelination or myelin sheath is very high in lipid content because the plasma membrane is high in lipid content. So it's going to appear white. White matter is going to be organized in tracts, as we've discussed previously. We have an ascending tract, which is going to carry information up towards the brain or up to superior parts of the spinal cord. And we have descending tracts, which are going to do the opposite, carry it to inferior portions of the spinal cord or to the effector organs. The white matter conveys impulses between the brain and the spinal cord as a communication center. And again, if you remember, the whole function of the spinal cord, white matter, gray matter, was to act as a relay station to transmit information from one section to the other. And again, in our diagram, you see that the gray matter is in the center. It's shaped sort of like an H, sort of like a bikini top. And in the center of the horizontal bit of the H, you'll see that there's a dark spot. That's that central canal we discussed. All right, that opening to allow for cerebrospinal fluid to be circulated. Around our gray matter is the white matter, right, that all those myelinated axons that we discussed. From the cord, we're going to find two pairs of projections, two pairs of sticky outies. We're going to have a dorsal root, and hopefully, obviously, that's going to be on the dorsal side of the spinal cord. When we talk about our dorsal root and conversely our ventral root, they're very specific, the information and the neurons that they're going to carry information from. The dorsal root conveys afferent nerves to the spinal cord. Afferent, hopefully we remember, is the sensory portion. So this is carrying information from all of our sensory receptors, our eyes, our ears, our pressure receptors in our blood vessels, our stretch receptors in our bladder, in our stomach, chemoreceptors in our blood vessels. All of that information is going to be sent through the dorsal root to the spinal cord. We're going to find in the dorsal root a dorsal root ganglion. Ganglion, we know, are cell bodies, neuron cell bodies. Specifically, it will be sensory cell bodies. The axons form the dorsal root and project into the gray matter. The axons from these sensory cell bodies that are located in the dorsal root ganglion. Okay? The afferent nerves meet other neurons, ascending and descending tracts. So those axons move into the gray matter of the spinal cord, and they're going to meet up with other neurons. They're going to meet up with the ascending or descending tracts. The ventral root is going to be on the opposite side, and that's going to be made up of efferent nerves conveying that information away from the spinal cord. That information will be going from the central nervous system out to our various effector organs, skeletal muscle, cardiac muscle, smooth muscle, and all of our different glands. The ventral root is formed by the axons of neurons whose cell bodies are in the gray matter of the spinal cord. So what we're saying is the root is going to be made up of axons. Where the cell body is going to depend on whether it's the dorsal root or the ventral root. If it's a dorsal root, the neuron cell bodies are going to be located in the dorsal root ganglion. If it's the ventral root, the cell bodies are going to be located in the gray matter of the spinal cord. Right? This is a really good diagram. You have this in your packet. You have this models, right? Photographs of models that look very much like this. You'll see the dorsal root ganglion, that kind of bulby looking piece. And again, just like our spinal enlargements, it's bigger in that area because we have cell bodies in there, not just strands. We have whole cells in there, so they take up more space. So that's going to be the dorsal root ganglion. And going from the dorsal root ganglion into the spinal cord is our dorsal root. That's made up of axons of those cell bodies that are located in the dorsal root ganglion. Conversely, we have our ventral root, right? That's made of axons that have their cell bodies in the gray matter of the spinal cord. So that's where that's coming from. When they're bound together, you have a spinal nerve, and you'll remember when we talked about our spinal nerves that there were 31 pair of those. Okay. Now we're on to the brain. We as human beings are kind of snobbish. When we think of the brain, we think of the cerebrum, the thinking part, if you will, the quote-unquote thinking part, because we're snobs, right? We think that's the most important part of the brain. Hopefully through this conversation today, you're going to realize that it's great and it's good, but it's not the most important thing, okay? Or the only important thing, we'll go that way. So the four main parts are the brainstem, the diencephalon, the cerebellum, right? Most of you probably have never heard of those. The ones that we focus on, and if you've taken a psych class, you really have focused on, is the cerebrum, that wrinkly part, because that's quote unquote most important to human beings. Now, when we go through our different areas of the brain, you'll see that that's not entirely completely correct, that there are other very important sections to the brain that may not allow us to think or feel, but they certainly keep us alive. So when we talk about protection for our brain, it's going to be surrounded by, enclosed by the bones of the cranial case. Okay, so let's think the parietal, the frontal, the occipital, okay, the sphenoid, throw the sphenoid in there. Also covered by the 3 layers of meninges, those same 3 layers of meninges that we talked about with the spinal cord. It's one continuous piece. So you may be thinking, well, Professor Kelly, then why do we divide it? We divide it simply because we talk about the spinal cord and the brain separately, so we have to talk about the protection separately to keep things straight, if you will. We also have cerebrospinal fluid. This circulates through the subarachnoid space, as we've mentioned, and the ventricles, and we mentioned that way back in the beginning of our lectures on the nervous system. And they're going to be spaces within your brain, okay? And those spaces within your brain allow for the circulation of cerebrospinal fluid. And it acts kind of as a shock absorber. Okay, so when you've got your brain in your skull and you hit a soccer ball, for instance, you bang your head against your nightstand, okay, you don't want your brain to be slamming right up against that bone, just like we had that fatty cushion for the spinal cord. For the brain, we rely more on cerebrospinal fluid. So if we look at this diagram, you're going to see the top diagram. We've got our skin, we've got our subcutaneous adipose, we've got the bone of our skull, right? We've got our dura mater, we've got our subarachnoid layer, our arachnoid layer, we've got our subarachnoid space. That's where we're going to have the cerebrospinal fluid circulating. Now, if you look in the lower diagram, you see those little black lines. Those are signifying, and you can see it in the top diagram a little bit better. This is another reason it's referred to as the arachnoid layer, because it's held in place. It's held elevated from the pia mater by those legs, and they look like spider legs. So that's where another reason, right? We said there are 2 reasons. This is the other reason we refer to the arachnoid layer as the arachnoid layer. We keep mentioning this cerebrospinal fluid. Well, where does it come from? It's produced by specialized sections or specialized areas within the brain called the choroid plexus or the choroid plexi, right, if there's more than one, that line the ventricles, and it's formed by filtering blood. It's going to keep any cellular material out of the cerebrospinal fluid. It's very much a watery solution of ions, glucose, right, all the kinds of things that the cells of the brain, the neurons, are going to need. And we get that by filtering the blood. And my cat just stepped on a key and changed the slide for me. It's going to be resorbed by the arachnoid villi and returned to the blood at the same rate. Okay, this is kind of a lot. Here's the deal. We're going to have the choroid plexi forming it. We want to have fresh cerebrospinal fluid, right? We talked about that. We don't want it, quote unquote, used. We need a fresh supply of oxygen, a fresh supply of glucose, of ions. The only way that we're going to be able to do that is if we replenish that. And if we don't remove some of that cerebrospinal fluid, then we're going to end up with too much fluid in these ventricles. So, as it's being formed by the choroid plexi, it's being drawn off by the arachnoid villi. Right? So we're keeping a finite amount. Let's say there's a cup of, and I'm making this up completely, there's a cup of cerebrospinal fluid. That has to stay constant because that's all the space we have for it. Because remember, our structures, our brain and our spinal cord are surrounded by bone. So we can't get too much in there or we're going to have pressure against the bone. So, as it's formed by the choroid plexi, it's going to be drawn off or taken off through the arachnoid villi. Now, this diagram you have in your textbook, and it kind of puzzles people, but it's really kind of a neat diagram. If you follow the arrows, that is the circulation pattern of the cerebrospinal fluid. The red areas in the center of the brain and around the cerebellum, those are the choroid plexi that are going to form the cerebrospinal fluid. And if you look up at the top, it looks like little blue mushrooms. Okay, those are going to be our arachnoid villi. And if we follow the arrows, it comes from the choroid plexus, circulates down and around the central canal, the arachnoid, the subarachnoid space of the spinal cord in the brain, and it's drawn off. If we follow those arrows, it's drawn off at those arachnoid villi. Those blue lines up top, those are representing veins, and the cerebrospinal fluid, as it's brought back out of it goes out of the subarachnoid space, is going to be drawn back into the circulatory system to veins contained within the brain. Now, what if the cerebrospinal fluid can't drain? Right? There's a blockage. Then the fluid is going to accumulate in those ventricles. And again, remember, you've got that skull surrounding it. Right? If my arm swells, the bone is on the inside, the swelling can get as big as it needs to. Right? It's not going to cause pressure on the deep tissue. However, with our skull, we have the bone on the outside. So if we're increasing the pressure inside, we're pushing the brain against the bone of the skull, and that is going to cause an increase in pressure inside of the brain. We call this internal hydrocephalus because the ventricles are deep inside of the brain, and we're causing that pressure buildup inside of the brain. Now, if we look at this poor little dude, he has an issue where the cerebrospinal fluid is accumulating in the subarachnoid space. This is referred to as external hydrocephalus because it's pushing from the outside in on the brain. This is very easily treated with a shunt from the ventricles. And a shunt is just a little teeny tiny tube going from the ventricles down into your jugular vein in the neck. And what you see in these photographs, on the left, I would say that this child is maybe 3-4 months old, and his skull is normal size, his eyes look great, you're just your basic little squishy child. On the right side, the pressure has started to build up. You see that his forehead is kind of enlarged, and his eyes are wonky. But again, this is going to be short term. This is going to be easily rectified by installing a shunt.